presentation that I'm very excited about, really a presenter I am very excited about, honor student, and most importantly, German minor, Aidan Engstrom is our final presenter in this room today. Not talking about German literature, but something from his other field, biology. His presentation is called Developing the Tools for Modifying the Genome of Cockroach-Infesting Parasites. He worked with Dr. Daniel Gold, who will come up to say a few things about him after the presentation, at which point we'll also have time for questions and answers with Aidan. And Dr. Gold will also be able to give you your cord and stole in recognition of your hard work. But without further ado, I want to turn things over to our final presenter of the day, Aidan Engstrom. Thank you so much.
So for my thesis project, I've been working on developing the tools to modify the genome of a cockroach-infecting parasite. So the parasites that we're interested in as a lab are called apicomplexans. They are a class of protists that are all parasitic. And you might be asking, why should we study apicomplexans? So most importantly, it's just that apicomplexans cause diseases. So if you look at this tree here of apicomplexans, some famous apicomplexans are Plasmodium. Plasmodium is the group of parasites that cause malaria. And then if you look here at this branch, there's Cryptosporidium. And Cryptosporidium is maybe less known than Plasmodium, but it is known to cause cryptosporidiosis, which is the second biggest cause of infant mortality due to diarrheal diseases. And so studying apicomplexans can help us better understand how to combat these diseases. So the parasite that we're most interested in, or the one that we're actually studying, is a gregarine. Gregarines are not really well understood, they're not highly studied, but they're a group of parasites that infect the intestines of almost every invertebrate on the planet. So our specific parasite is called Glabrocholomicroter. One interesting thing about gregarines in general is that they lead an epicellular lifestyle. When you think of a parasite, usually they'll either be extracellular, where they're living outside of the cells in their host, or intracellular, where they're living inside the cells of their host. But here, an epicellular parasite will sort of be in between, where they will invade a portion of the host, but not the entire host. So additionally, gregarines have high host specificity. There's no danger in when we're studying Glabrocholomicroter getting infected ourselves. And finally, they have easy to care for hosts. What are their hosts? They're Madagascar hissing cockroaches. So here you can see a Madagascar hissing cockroach, and one thing that's very helpful in our research is that Glabrocholomicroter is the only gregarine that we know that will infect our host. So there's no chance of finding another gregarine inside of our cockroaches. Additionally, it leads a simple fecal oral lifestyle. There are many different complex life cycles that parasites can lead, and this is a fairly simple one. And then finally, we have large hosts. These cockroaches get really big, so they're pretty easy to dissect compared to another, like a smaller one, like a fly or something like that. And additionally, the parasites are really big. Here you can see inside the intestine, they can get up to about 1 millimeter in length. So let's look more at the life cycle of our parasite. So when the cockroach in the beginning eats our parasite, it will be inside these oocysts. And when the oocysts reach the intestines of the parasite, small cells called sporozoids will emerge from the oocyst and epicellularly attach themselves to the epithelial lining of the cockroach's intestines. Following that, they will develop and reproduce and produce a gametocyst, and then the cockroach will pass the gametocyst in their feces. And this is what the gametocysts themselves look like. They're small and pearlescent, but they're easy to see with your eyes and you can pick them out. And then inside, theoretically anyways, these cockroaches are in the rainforest of Madagascar, which is a very hot and humid environment. And so when it's sufficiently hot and humid, these gametocysts will sort of explode and shoot out these long chains right here of oocysts in a process called dehiscence. And looking at these chains under a microscope, you can actually see these are the oocysts, and inside each oocyst is 2 sporozoites. So that's a general idea of what our parasite is doing in their life cycle. So part of our research in understanding this parasite would be aided in having the ability to modify the genes of the parasite. You know, you can either knock out genes inside the parasite in order to determine what those genes are necessary for. Additionally, you can add protein tags onto different genes inside the parasite to see what those genes are producing, where they're being localized, and things like that. But the problem is that no one's ever actually been able to put transgenes into a graven before. No one's been able to create a transgenic parasite. So my goal was to develop a model and a method for modifying the genome of laboratory migratory organisms by inserting genes for reporter proteins into the parasite. So here is the basic plan that I set out to do when I was doing this project. First, I'm going to be inserting the genes inside of sporozoite of our parasite through a method called electroporation. After that, with my new transgenic parasite, I'm going to be injecting that transgenic parasite into the cockroach. And then finally, I'll be dissecting the cockroach and looking to see if I can see the parasite expressing those reporter proteins. So you might be wondering why I am injecting the parasites through a syringe, it looks like. So in the past, in past research that I've done, I've found, in past research that we've done in this lab, a former honor student, Maureen Padilla, found that feeding cockroaches paramomycin, which is an antibiotic, will lead to reduced levels of parasites. The problem with that is that the method of delivery for the paramomycin was just sprinkling it on some carrots, which the cockroaches like to eat, and you could never give a reliable dose. And so my work was finding that when you actually inject the cockroach with a syringe, you're able, in this case I was injecting blue food dye, you're able to see that the injected solution inside the intestines of the cockroach. So I said earlier that I was going to be using reporter proteins. So let's talk about that. Our parasite doesn't normally express bioluminescence. That would be weird if there's no reason for it to. And so if I took bioluminescent proteins and put them into the genome of the parasite, I would know with 100% confidence, basically, that those were from the genes that I inserted into the parasite. So if you look right here, these are the 2 genes. This is just sort of a cartoon of a DNA fragment that I'm using. The first protein that I was looking at was nanoluciferase, which is similar to the bioluminescence in fireflies. When it's exposed to a chemical, it will produce light. And then also I'm looking at amnion gridage, which is another protein that is bioluminescent. It's a form of GFP, which was originally found in jellyfish. And then if you see here, there's this P2A. That's just a self-cleaving peptide, so that when the parasite is expressing these 2 genes at the same time, the P2A will cut the protein, so it's 2 separate fragments rather than 1 large fragment, so that both of these proteins can work as a whole. And normally when you are producing transgenic organisms, you would insert your transgenes, replacing genes that you would think are redundant. But the problem with that is that we don't know, we can't confidently say which genes of our parasite are redundant. And so we have decided to, we're looking at, we chose a specific protein called beta-actin, which is a cytoskeleton protein. You see it in a lot of eukaryotic, it's a very highly conserved protein that you see in a lot of eukaryotes. And the most important thing is that we are assuming that it will be expressed at all stages of the parasite's life cycle. And so what we've decided to do is we're going to copy the promoter and 3' UTR of that gene and place the R2 transgenes in between those two sequences. And then finally, if we're not going to be replacing a redundant gene, where are we going to put the transgene? So if you see here, this is a map of one of the largest contigs we have of Blabbericula migrator's genome. And we chose this site right here because it's far away from any potential genes that we've identified using bioinformatics. And so these two DNA sequences here are the upstream and downstream sites of where we're going to be inserting our transgene. So I have all of these sequences here, but they're not really connected, so how do I put them together? Well, if you notice here, all of the colors match, and that's sort of symbolically representing that when I'm amplifying these fragments using PCR, I'm adding, say, the beginning of nanoluciferase to the end of this promoter sequence, and so that they match, and they'll be able to line up like this. And when I have these matching sequences, I'm able to assemble these fragments that are unrelated at first together into one larger fragment using Gibson assembly. So I could, once I make this sequence once, I could just use it as is, but Gibson assembly and the PCR is very expensive, both time-wise and reagent-wise. It gets expensive very quickly. And so one other form of attack is considering plasmids. Plasmids are just small circular rings of DNA that bacteria will use. And if I insert my sequence here into a plasmid, I will be able to use E. coli and insert the plasmid into the E. coli and have them do the work for me and amplify this sequence from just having a few molecules of it to having millions. So we're going to look at what my successes have been so far. If you look at these two transgenes, the nanoluciferase and the amnion green, they have an expected length of 605 and 731 respectively. You can see that I get very thick bands when I separate the DNA by size using gel electrophoresis, and I don't get any other fragments from my PCR. This is because I'm using plasmids to get these genes, because that's where I ordered them from. But on the other hand, the other sequences are using our parasite's DNA, our parasite's genome as a template, and that's a much more complicated template for PCR. And as a result, if you look here, I'm using two different samples here. This is our promoter. It's expected to be 2,000 base pairs long, and you can see a very faint band here, but this is all I have for it. There's not, I haven't been able to produce anything else like as robust as this. And then looking here at the 3' UTR, this is supposed to be 290 base pairs, and that is what it is. And then finally, looking at the 3' target site, I've been able to produce that. At the 5' target site, I have not. This is the one that I'm having trouble with. So if you look at this map of my work, I have had successes in all of these fragments except for the 5' target site. Following production of the 5' target site, I will assemble this plasmid using Gibson assembly and amplify it using E. coli bacteria. And I will insert that electroporate sporozoites and inject them into the parasite, and I will hopefully be successful in producing a model for how to produce transgenic parasites. And this is where I'm at so far with my project. So I'd like to thank everyone in my current lab, everyone in Dr. House's lab, Emory. The bioinformatics work you guys have done has been very invaluable for my project. Additionally, Dr. Gold, Hannah Trogdon, my parents, the honors program, and Dr. Griffin. Thank you so much.
Thank you so much, Aidan. Before we get to questions, I want to invite up your advisor, Dr. Gold, to say a few things about working with you and also to present you with the cord and the stole that you'll wear at graduation to represent not just this project, but all of the hard work we've put into the honors program and to your work here at St. X.
So it's one of, I think, the best parts of my job is sort of seeing students grow, right? And so, Aidan started working with me as a research student, your first semester of sophomore year? And so, he's a senior graduating now, so I've gotten to see, 3 years of Aidan's growth. And the thing about Aidan is he was already pretty high level when he started, right? He was, things I look for in students are they dependable? And I can tell you that Aidan is easily one of the most dependable students I've ever had, to the point where, I know she couldn't hear you today, but Zoe Skelton, the department's lab coordinator, Aidan was always her go-to student for anything she knew that she needed to get done and get done right and get done well. And I think that's always been true, as well. And also, Aidan, I've had him as a student in multiple classes, and just to see his growth as a scientist has been, just a real pleasure. And also, he's an interesting person. He's got a lot of deep thoughts about a lot of things, and he's somebody I enjoy talking about with. And the project that he's worked on multiple projects, and this is just actually the most recent one. And it's a project that I've been wanting to have done in the lab for quite some time, but I've been kind of waiting for the right student who had the right set of interests, the right set of skills, and the right. It's really easy. It's an experiment. I told him this when he began. This isn't going to work the first 5. I think I told you. Don't expect this to work like the first 5 times, right? This is one that's going to be you're going to have to really some of these might be easy and some of them were, but some of them are going to take you some time. And I didn't want to give it to a student who I knew okay, it doesn't work the first time, and they kind of shut down, and that's going to be it, and I have to kind of feel like I knew that was enough to work with. I knew Aidan would be mature enough, and he would be a good enough scientist to say, okay, what did we get right? What did we get wrong? What can I do? I can go back and just do it and redo it, and he's almost done. I mean, really, in terms of what I think is the difficult part of the project. And so I was really just waiting for somebody who I knew it's a pretty tricky project to handle. And this is just one of many. I'm just so impressed with Aidan. It's one of those things also that's, from a purely selfish perspective, your best students, they're great, and then they graduate. So I'm really going to miss Aidan, on multiple levels, both for what he contributes to the personality and the camaraderie of our lab group, and just as what a great scientist he's already become, and I know that he has growth in the future, that he'll even be better. So I'm very excited about that. So how do I do this road for skull thing? I've done it's been a while. My honor students have all recently, it's been actually a few years, most of my honor students have been virtual.
Exactly, we're going to pretend with a computer.
There you go.
Congratulations. Questions for Aidan?
Yeah.
Can you get more detail on how you get the plasmid part that you need when you go on and get that into the parasite itself?
Oh yeah. So when you can electroporate the E. coli in the same way that you electroporate the, as I plan to electroporate the sporozoa. And so you just, I don't know exactly how it works. I haven't inserted a plasmid into the E. coli yet, but you just shock basically with electricity the E. coli and mix it with the plasmid, and some of the plasmid will end up in some of the E. coli as the media. And then when you grow that E. coli on a plate, then you can pick out colonies that have been transformed. Here, let me actually, I've got a slide somewhere, right here. Yeah, so you're able to pick out what has been transformed based on this ampicillin resistance here. Only the transformed bacteria will survive. And then you can actually just centrifuge down the surviving colonies that have been transformed, and from there, you will just isolate the DNA and pick out nothing. Yes?
Can you describe a potential application of being able to edit the genome of the parasite?
Yeah. So, for example, if we locate a specific gene that we're interested in and we want to know how it functions and what it is doing for the life cycle of the parasite, then we can go into the parasite and just cut that part out, and then see how it's functioning after it's missing that gene, see how that has affected its growth, stuff like that. That's one example of understanding the genes.
And so what you're developing here would allow you could replicate this essentially with any gene.
Yes, so this is a proof of concept to show that, we understand, like, the genome that we have is correct and stuff like that. It's a proof of concept that electroporating the sporozoa will work and stuff like that.
I feel a little like I'm in an alternate universe because I know you obviously as just a super brilliant German student and critical thinker about literature and history, and it's so cool and a little bit tricky to see you talk to an alternate about something so totally different. But to the extent that I followed it, it's really, really fascinating. Last question for Aidan.
Yes.
I'm not familiar with the Gibson methodology.
Yeah.
How does that work?
So, I've got a slide. Yeah. So, like I said, these two fragment A and B, for example, have an overlapping sequence. And so when you are mixing it with just a concoction of different DNA repair enzymes, so this 5' exonuclease will chew back to this, and these two DNA strands will be homologous, and some of them will bind together, and you can use DNA polymerase to extend and fix this up, and then DNA ligase to seal the nick, and it just puts the two molecules together.
Kind of similar to a restriction enzyme cut site.
Yes, yes.
You're using exonuclease instead.
Yeah, so it's like a restriction enzyme, but not restricted because you can use any sequence that you want.
That's really cool. Because then I know there's like a weird sequence in the middle. So that makes the reason.
Yeah.
It's also trying to overlap 5 different things, trying to find 5 restrictions.
Yeah,
that
that seems like a pain.
it would be a pain, so you could just, all you have to have is overlap.
That's really cool. I never seen
Really?
No,
No,
I always
it's-
just use restriction cut sites.
Yeah, no, no, Gibson is, once you go Gibson, you don't go back.
Yeah.
Once you go Gibson, you don't go back. And all this stuff is like, no, I mean, nothing is new, it's just like,
So, gratuliere, danke für den Vortrag, das war hervorragend. Congratulations.